right, welcome back to Chemistry 4110. We're hitting the home stretch. Today we're going to talk about part 3D, which is sugars and nucleic acids. And we're going to be talking specifically about something called aptamer design and selection. So let's get started. All right, aptamer design selection. All right, so I want you to think and recall the peptoid screening libraries that we learned from the last unit. So peptides and proteins, and remember when we had large libraries of peptides that we could generate. This was very powerful because we could generate, potentially millions of different unique compounds simply by altering the number of different R groups. So if this was a very successful way to develop novel compounds that can bind to specific targets, could we also design DNA or RNA sequences that also bind to specific targets? So this is actually a technique called SELEX, and it actually stands for the systematic evolution of ligands by exponential enrichment. And the way that's going to work is similar to peptoid libraries or peptide libraries, where you'd have a large number of different compounds. And so the same application is going to apply for nucleic acids. Imagine you have a large pool of structures. You're going to add your target, and then you're going to ask which compounds, which nucleic acids bind with high affinity and high specificity. So here you can see that some of them will bind, but most of them will get washed off and discarded. And then you'll take your compounds that bind, you'll elute them off with maybe some temperature or high salt. And then what you'll do is you will take these sequences and you'll re-amplify them to further optimize a secondary pool that could further strengthen binding to these targets. So my question is, how do we initiate that first pool of libraries that can bind to the target? So you could think of, okay, we could make DNA or RNA individually and then mix them together. That could be very tedious though. So instead, what researchers can do is they can say, okay, we have 4 different bases, and N here is going to be equal to 1 of the 4 bases. And if you randomize, for example, with one of the 4 bases at 1, 2, 3, 4, 5, 6, 7, 8 different positions, how many different DNA sequences can you generate? So it turns out if you go 4 to the power of 8, that is around 65,000 different DNA sequences. So this is quite intriguing. Now, this concept of SELEX was actually developed by 2 scientists, Andrew Ellington and Jack Stozak, and they published a paper in 1990 in Nature, which is a big journal in science. And this was the first paper describing that you can actually use nucleic acids through the selection to make molecules to find what we call sequences that bind high affinity to targets. Just a word about Jack Stozak. He actually is a Canadian-American chemist who actually grew up in Montreal and Ottawa. He did his bachelor's at McGill and then later did his PhD at Columbia in New York. So what else can I tell you about this? So once you develop and once you figure out which ones bind and you elute them off, what you do is you amplify them and then you put them into like an expression plasmid vector, right? And then you can express those to further amplify the gene pool. Later you can evaluate which ones are binding based by simple sequencing, and that's definitely a lot easier now than it was in the 1990s. I just want to show you how efficient this is at binding to specific targets. So I'm going to draw for you two molecules that will look very similar, and I'm sure many of you have ingested these molecules before. Don't worry, they are not illegal. So the molecule on the right is something that you might find at your local Starbucks. So this is our good friend caffeine. And on the left is something called theophylline. Theophylline is a component in things like chocolate and tea, and caffeine, of course, is in coffee. And they both, they both increase alertness, and they both have, if you're a student, which you are, they can have very positive effects. The only difference between caffeine and theophylline is this nitrogen here has a methyl group on it, whereas in theophylline you just have a proton. So there was a study done in Science in 1994 in which they wanted to see how specific they could get these nucleic acid aptamers binding. And they wanted to see whether they could differentiate between an aptamer binding to caffeine versus theophylline. And so they did a very similar type of selection experiment. I forget exactly how many number of bases they used, but what they noticed was that they developed an aptamer that could bind 10,000 times stronger binding affinity to caffeine compared to theophylline. Okay, so we're just talking about a single methyl group here. So you can actually use this to detect small changes in structure, and this could be very important for metabolites, biosensing applications, if you're trying to determine, not just small molecules, but any small structural changes and perhaps large biomacromolecules. So 10,000-fold change in binding affinity for caffeine compared to theophylline. So that was published in Science in 1994. I'll give you sort of the coordinates here. Volume 263, page number 1425. So SELEX has been around, and these adopt very concrete three-dimensional structures that can bind to its target. Here I'm showing you a small molecule target. So there's a company called Optigen. It's in the US, and they on their webpage they say, well, forget antibodies, use aptamers, because aptamers could be used to bind to potentially any molecule, and antibodies are specific proteins that bind to specific molecules or other proteins. And here you can see a large oligonucleotide synthesis machine in which you could generate large libraries of these. And here's an example of an aptamer that binds against vitamin B12. And you can see here that this aptamer has a very specific three-dimensional shape. And they claim that they can have fast, inexpensive, and as simple as ordering an oligo from your favorite supplier. So definitely check out Optigen on your Google list. So are there any commercially available aptamers as therapeutics? Well, there are only a couple. Okay, so there's one that's called Machogen, and this targets a protein called VEGF, which stands for vascular epithelial growth factor. And this is it's a growth factor that promotes the rapid division and growth of cells, but it's also, or can be, overexpressed in a lot of cancer cells and tissues. So the idea behind Machogen is that it would literally bind to this protein and prevent its function, or prevent that protein from functioning inside the cell. So what does Machogen actually look like? So Machogen's two-dimensional structure is shown here, and this was approved in 2004. And let's take a look at some of the molecular components of this molecule here. So you can see here that You have some fluorinated groups. Here you have O-methyl groups. Okay, so we're looking at sugar modifications that improve the stability and the specificity of the molecule. These adopt C3' endo sugar units, which we've discussed already a little bit. And you can see here that they have this interplay between fluorinated and O-methyl. I'm not exactly sure why or how they decided on which combination worked, but this is the one that works for Optigen the best. Some other things that I just want to note is that they have attached on the 5' end here, they have this CH2, with 5 carbons and then a nitrogen here. So maybe I'll just kind of draw that in sort of chemical structure orientation, and this looks like this. And under physiological pH, a nitrogen would be protonated, and that secondary, or that protonated nitrogen makes it positively charged, which could really help it perhaps with cell membrane delivery. Because we're modifying the sugars here, this is classified as a second generation chemically modified oligonucleotide. And of course, it doesn't have a sort of a two-dimensional structure as I'm showing you here, but it would have a very unique three-dimensional structure that's able to bind to VEGF. Okay, so that is Machogen. And I really just wanted to highlight one example here, just to give you an idea of what SELEX and what nucleic acids can do in terms of screening. Now, I just want to highlight some examples of, or some comparisons of using antibodies versus aptamers. Okay, so we haven't talked about antibodies, but let's sort of compare their pros and cons. Let's start with antibodies. So I know a lot of students here may not know exactly how antibodies are raised and developed. Essentially, to raise an antibody, whether it's for pharmaceutical development or whether it's for a research tool in the lab, typically you need to use an animal. So sometimes you might use a rabbit or a bovine, which is a cow, or sheep or goat, and you literally inject that animal with your target of interest. The animal will develop antibodies to it, and then after sufficient antibodies are made, then you have to sacrifice the animal, collect the antibodies, and then you have to purify them and store them at 4 degrees. So there are some significant disadvantages with antibodies. One of them is you could think of the ethics and the costs involved with doing that. You really don't get that much antibody from the animal. And of course, there's the whole ethics component as well, right? Is it ethically responsible to sacrifice an animal to generate antibodies? These antibodies are proteins, and these proteins will unfold and denatured if left at room temperature. So these antibodies are easily degraded. They need to be stored at 4 degrees in the refrigerator, and they have a very finite lifetime. So they can't be stored indefinitely. And as I mentioned, animals need to raise antibodies. Now, antibodies have been, for a long time, been perceived as being the #1 type of molecule that can achieve specificity. I'll give you an example. So antibodies for specific proteins can be made, and if you have a single phosphate group on a protein, you can develop antibodies that distinguishes between a phosphorylated protein versus an unphosphorylated protein. So that's a very small change in structure, yet you can develop an antibody that can differentiate between that. Now think back to that caffeine theophylline experiment that I talked about. That was a proof of concept experiment describing that you can now generate an aptamer nucleic acid that could achieve perhaps that same level of specificity. So there's certainly a lot of promise there, although there are some skeptics who say that maybe you will not be able to get that same high, super high level of specificity. 10,000-fold change in binding affinity seems pretty good to me, but who knows what the future will say. For aptamers, well, what do you need? You need a DNA RNA synthesizer or access to libraries of DNA RNA. And today in 2020, it's actually not too difficult to do that. You can perhaps purchase some DNA libraries at a fairly low cost. In general, these would be faster to make and to select, and you don't have to use animals, obviously, to raise an aptamer. And furthermore, one of the nice features of nucleic acids is that they are relatively stable on their own. So you could potentially develop an aptamer, and you should be able to store this at, minus 20 or even at room temperature. And a lot of them should be able to fold reversibly. So you should be able to heat it up and then slowly cool it down so it can form its unique three-dimensional structure in order for it to bind. Okay, so that's the brief lecture today on aptamers and SELEX. All right, see you soon.